Okay, as Kiri has alluded, we're going to cover natural and artificial metal enzymes today in this class. I'm going to start by definition of the term itself, and then just a quick overview of the types of cofactors that nature uses for metal enzymes. So broadly speaking, metal enzymes would be defined as enzymes that contain metal cofactors, and the metal can either be bound directly to different residues within the protein structure, or bound to the protein structures through a prosthetic group such as porphyrins. This slide shows an overview of, this is not exhaustive, just select overview of the types of cofactors that nature uses for Metalloenzyme catalysis. On the left side, you'll see different types of oxygenases that we would cover in class. Most of you probably would be familiar with the P450s, which is the most well-studied enzyme within this superfamily. In general, all of these oxygenases catalyze oxygen transfer. And just a quick definition, there's monooxygenase and dioxygenase. For monooxygenase, only one oxygen atom from dioxygen would get transferred into the substrate, whereas for dioxygenase, both oxygen atoms from dioxygen would be incorporated into the product in the transformation. I'll quickly draw your attention to methane monooxygenase, which is an enzyme that's capable of hydroxylating methane to methanol. On the right-hand portion of this slide, you can see several more exotic cofactors. We'll cover a little bit cover 4-iron 4-sulfur cluster a little bit later. There's also ribonucleotide reductase, which has some structural similarity to methane monooxygenases. So ribonucleotide reductase is responsible for the conversion of ribonucleotide to deoxyribonucleotide, so essentially conversion of your RNA to DNA. And this enzyme family has been extensively studied by Joanne Stubbe, and I think she essentially has made a whole career out of it. And then lastly, there are, of course, many exotic cofactors that nature has employed for catalysis. You can see there's that iron-iron hydrogenase that's responsible for the reversible conversion of hydrogen to protons and electrons. There are some enzymes that contain molybdenum, such as xanthine oxidase. This enzyme catalyzes the oxidation of hypoxanthine to xanthine. And recently, I think a couple of years ago, there was a big discovery of an enzyme that actually contains a nickel pincer as a cofactor. This enzyme is responsible for the racemization of lactate, so the S and R isomers of lactate gets interconverted, and of course there's the FIMOCO hydrogenase, which is responsible for the conversion of hydrogen to ammonia and has really wild-looking gauge cofactor, as you can see, that also contains multiple iron, multiple sulfur, and one molybdenum site. Then I thought I will also give a quick overview of the types of tools that you can use to study metalloenzymes. And there are many different types of spectroscopies that you can use to study metalloenzymes, and these take advantage of different types of excitation mode of your metal center, essentially. So you can see from this slide that the types of energy gap that you can try to excite can vary quite greatly, ranging from probably the most highest energy, the gamma ray. This would be employed in most power spectroscopy, and what it does is essentially it essentially excites the atomic nucleus, and and you can detect various changes in electronic state by doing this excitation. And you can go all the way up in the energy level to things like UV vis or magnetic circular dichroism, which allows you to detect different types of electronic transitions within the Metalloenzymes, and then go all the way to things that are much lower in energy, such as microwave and radio wave. In this case, it would manifest itself in the form of EPR, which studies the transition between the different electronic spin states of your metal center. So just be aware that all of these techniques exist, and for the purpose of this class, we won't go into too much details of how they work. Okay, we're going to cover oxygenases first. We'll start with the most well-studied one, the P450s. The P450s they all share the same protoporphyrin IX cofactor that I have drawn, whose structure I've drawn on the top left-hand portion of the slide. In general, the enzyme superfamily itself shares pretty similar fold that has pretty high content of alpha-helices, as you can see from the crystal structure that I've put up there. All P450s conserve cysteine that ligates the iron center at the axial position. This exists as the cysteinate, and the thought is that the tilate ligand actually acts as a good electron donor that will give a push effect for dioxygen cleavage, as we would see in the catalytic cycle later. You can easily detect AP450 by incubating it with carbon monoxide. This would give and then reduce the iron 3 species to iron 2, and this would give a ferrous CO complex that has a characteristic big absorption peak at 450 nanometer if you run a UV vis. However, one of the most important things to note about the P450s is that they require several components to be fully functional, and the other components are required to deliver electrons to the iron center. There are several different types of electron delivery system, as I've highlighted in the bottom left portion of that handout. I think the earliest one that was discovered is the class 1 P450 that has a separate iron-sulfur domain and FAD domain. So FAD is flavin adenine dinucleotide. We will see the structure of this compound in the next slide. And in this system, the FAD domain would consume a molecule of NADH or NADPH, and this allows the FAD domain to deliver electrons to the iron-sulfur cluster domain that in turn would then deliver the electron to the heme domain before catalysis to happen. There are also many P450s that belong to the class 2. In this case, they don't contain any iron-sulfur protein, and instead they have two separate flavin-containing proteins in the form of FAD and FMN domains. These are both flavins that have slightly different types of side chain, as you will see in the next slide. Most of these systems exist as separate proteins, with the exception of a few. There are some P450s that have fusion between the heme domain and FMN and FAD domain, and this actually allows a more efficient electron delivery system because they're essentially connected together. And I should also point out that there are other classes of P450s and new electron transfer chain mechanisms that recently been discovered also. Okay, so just to quickly going through the catalytic cycle of the enzyme, we'll start from the top here, which is the ferric form of the protein. Notice that it has water ligation at this stage. So the first event that happens is substrate binding. This would displace water molecule from the iron, and while this is happening, there's also a spin shift that happens in the iron itself. So it goes from low spin to high spin, And through this change in spin state, it's been observed that the redox potential of the iron itself becomes more positive by about 100 millivolts, and this makes it easier to reduce to iron 2. So essentially, substrate binding triggers the reduction event of the iron center to iron 2. So the reduction happens through delivery of single electron from the FAD and FMN domain. And then at this point, we can go to the oxygen activation step. So the ferrous species would first react with dioxygen to generate a ferric superoxo species down here. And then another electron delivery coupled with a proton transfer would generate the following ferric hydroperoxo species that's also commonly called compound 0. And this species can then undergo a protonation that will lead to cleavage of the OO bond, and this is where the thiolate ligand actually also assists in giving a push effect for the cleavage of the OO bond. And this results in the formation of an iron oxo species that is also a cation radical character. This compound is called compound 1, and the identity of this species was characterized by Michael Green in this science paper in 2010. And this is the species that's responsible for your oxygen transfer, essentially. So the first thing that happens is hydrogen atom abstraction from your substrate to give the following species that's called compound 2. And then there will be a radical rebound event that then gives you your hydroxylate product, and then return the metal center to the ground state again. And if you're interested in more details about the elementary steps in this catalytic cycle, you can refer to all these reviews here. But I thought we'll also go through a little bit more details on the electron delivery system of the P450s. So shown on the top left is the structure of FMN and FAD. So they both contain flavin, which is this heterocyclic molecule over here, but they have different types of side chain where FMN is a mononucleotide. Sorry, there's actually a mistake in the slide. This should be a diphosphate bridge between the nucleobase and the FMN. And just to highlight how the electron transfer cycle would happen, I thought I would illustrate this point by using E450DM3, which is probably the most well-studied enzyme within this superfamily. And this was based on a study that was performed in this biochemistry paper in 1996. So the first thing that happens is binding of NADPH into the FAD domain, and then there will be a hydride transfer that happens between NADPH and the FAD cofactor in the FAD domain. And after this hydride transfer, there is the reduced NADP plus would then be replaced by another molecule of NADPH, and at the same time, the reduced FADH would then perform a single electron reduction of the FMN in the FMN domain of the enzyme. So this would lead to formation of FMN semiquinone, whose structure is shown up here. And then instead of getting reduced all the way to the hydroquinone, at this point, the FMN semiquinone would actually do an electron transfer to the ion center to reduce it to iron 2. And then another single electron transfer happens from the FAD to FMN to again regenerate the FMN semiquinone. This would then deliver the second electron during your oxygen activation step. So I think the take-home point is that there's sequential electron transfer, and it has to be carefully choreographed to couple the electron delivery system and oxygen activation for productive catalysis without leading to undesired side reactions with your ion center. And it's actually been proposed that the FMN hydroquinone species here is incapable of reducing iron. And then I thought I would also highlight the protein dynamics of electron transfer. So it's been proposed that the FMN domain for P450PM3 actually lies about more than 10 angstroms away from the heme center. So it's pretty far away, and people are trying to figure out how the electron transfer can happen very efficiently. And it's been proposed that the FMN domain itself can actually undergo a large-scale movement through this flexible hinge region that would bring the FMN domain closer to the heme domain to facilitate a more efficient electron transfer. Now we'll move on to the types of catalysis that can be performed by the P450s. They all belong to oxidative transformations, and this can range from your conventional hydroxylation reaction to desaturation. This would happen through formation of your cation followed by proton elimination. There is also one P450 that has been identified to catalyze hydration. Of course, it can also do other types of oxygen atom transfer, such as oxidation or aromatic hydroxylation, as well as N-hydroxylation. And there are also some P450s that can catalyze biaryl coupling. This would happen through hydrogen atom attraction of your phenol, and then the resulting phenol radical can then undergo a biaryl coupling. And it's been found that different P450s can utilize different intermediates in the catalytic cycle for catalysis. So the active species is not confined to just compound 1. So for example, it's been, in some cases, it's been proposed that compound 0, which is the following hydroperoxyl species, is the active species for epoxidation. And then in particular for aromatic nitration, which was discussed in this Nature Chem Bio paper in 2012 for the biosynthesis of taxotomin. It's been proposed that the active species in the reaction is a ferric superoxide that can then react with this hydroxyl free radical to generate a ferric peroxynitride species. This can then undergo hemolysis to get to compound 2 and the following active nitrating agent that can then do nitration of molecule of tryptophan that is found in one of the fragments in taxonomin. P450BM3 is probably the most extensively studied members of the P450s, mainly due to the fused nature of the enzyme. So the FAD and FMN domain are fused together with the heme domain, and this makes it a lot easier to work with. And at this point, I think studies on the P450s as well as their applications in catalysis have been pioneered by Francis Arnold, Manfred Reeds, and several other people like Louet Long in the UK. And I think hundreds and hundreds of mutants have been generated at this point, and based on this performance of these mutants, people can start figuring out the roles of different residues within the proteins. This slide also gives an overview of the native function and of the enzyme, which is to perform hydroxylation of long-chain fatty acid to give a mixture of hydroxylated product. But by engineering of the wild-type enzymes, many labs have discovered that you can perform site-selective hydroxylation of different types of scaffolds that can potentially be useful for synthesis and so on. And I guess in this slide I've been mostly focused on highlighting selective hydroxylation of terpenes. That's probably really hard to perform using your traditional small molecule catalyst. I should also point out that the crystal structures of P450BM3 and the reductase domains have also been solved independently, and this also really assists in engineering efforts of this enzyme to change its substrate selectivity profile. Okay, we'll move on to a different family of oxygenase. In this case, we'll cover a little bit the iron and alpha-ketoglutarate dependent dioxygenase. And this enzyme superfamily shares a common 2-histidine and 1-carboxylate iron binding motif. And it also contains a ketoglutaric acid co-substrate that is consumed stoichiometrically in the reaction. And the ketoglutaric acid co-substrate binds in this mode to the iron. In the catalytic cycle, substrate binding would displace the water molecule from the active site, and this would also trigger several steps for dioxygen activation. I think the interesting part here is that the dioxygen activation is coupled with a decarboxylation process that also converts ketoglutaric acid to succinate. And this happens through the formation of the following bicyclic species that happens through attack by the superoxo group on the carbonyl, and then you can push electrons to decompose this, and that will lead to generation of iron oxo species as well as loss of CO2. And once you get to the iron oxo species, the remaining steps is quite identical to the P450s, so you will see a hydrogen atom abstraction followed by radical recombination to give you your hydroxylate product. And at the end of the reaction, you lose your succinate byproduct, and then molecule of alpha KG would then complex to the iron once again and return to the catalytic cycle. The first discovery of, so I'll just call it alpha KG for now, so it's a bit easier for me. The first discovery of members of this superfamily was actually reported in 1966 in the form of the discovery of a prolyl hydroxylase, and this enzyme catalyzes selective C4 hydroxylation of prolines, and some of these hydroxyproline residues can actually be found in collagens, and it's been suggested as well as verified, I guess, that hydroxyprolines are important components for collagen so that it can form the supercoiled motif. On this slide, I've also highlighted the types of hydroxylation reaction that can be catalyzed by alpha KGs, and this can range from hydroxylation of free amino acids to selective hydroxylation of indole alkaloid. In this case, this belongs to the phenolene biosynthesis, as well as terpenes. In this case, I've highlighted the work by our colleague in the Florida campus, Ben Shen, in the identification of an alpha KG enzyme within the biosynthesis of potentiamycin. I also would like to highlight different type of reaction that can be catalyzed by the alpha KGs. So there are actually a small subset of the alpha KGs that can perform halogenation. And in this case, the iron binding motif is a little different. So instead of a carboxylate, you actually would have an alanine or a glycine in the active site. So this free up one coordination space, and this would be occupied by a chloride group. And in the mechanism for halogenation, after hydrogen atom abstraction, the resulting radical species would selectively undergo radical recombination with the chloride group instead of the hydroxide. And studies by Marty Bollinger and colleagues have suggested that the enzyme active site plays an important role in positioning the substrate such that the resulting carbon radical would actually be found a lot closer to the chloride instead of the hydroxide, and this preferentially leads to halogenation reaction. The first characterization of alpha KG halogenase was done by, I think this is a collaboration between the Bollinger group and the Walsh group, and this is found in the biosynthesis of syringomycin, where it was found that there's an enzyme called SIRB2 that can catalyze chlorination of the terminal metal group of a PCB-bound threonine, and this amino acid itself can be found as one of the fragments in syringomycin. And I should also mention that the Bollinger-Krebs group at Penn State have done a lot of amazing work in understanding the mechanism of this enzyme CRB2. And they have also shown that under stoichiometric conditions, you can actually perform radical nitration and acetation by using CRB2. Although the turnover is not great, so this here they actually do a one-to-one stoichiometry between the substrate and the enzyme, and they're getting less than 50% yield. So essentially, you're not getting a full turnover essentially in the reaction. So I've shown an enzyme that an alpha-ketoglutarate halogenase that can catalyze halogenation of substrate that's bound to a peptidyl carrier protein. But recently, there's a standalone alpha-ketoglutarate halogenase that was characterized that can act on a freestanding substrate that's not bound to a carrier protein. And this can be found in the biosynthesis of the low-beta-nolinones, an enzyme called WEL-O5, and another homolog called AM-O5 can catalyze CH halogenation at this carbon here to give the corresponding chlorinated product. And in this paper in 2016 by Yushin Yu at Pittsburgh have shown that you can do some type of fusion protein to alter the future selectivity of the chlorination reaction itself. Now, you might think that on first pass it might be easy to convert a hydroxylase to halogenase by simply taking out that carboxylate ligand, replacing it with something that cannot bind, and then add a halide to the reaction. But this is not the case. You cannot simply convert a hydroxylase to halogenase. This is a work that was, I think, studied pretty extensively by Ron Raines in this +1 paper, where they show that if you just simply mutate the carboxylate to alanine or glycine, you would actually get a non-functional enzyme that cannot do either hydroxylation or chlorination. And I think in this regard, in terms of swapping hydroxylase to halogenase, I think the most success, Amy Bowles' group at Penn State probably have achieved the most success, as reported in this Nature Chem Bio paper, where they were able to get crystal structure of the enzyme, and by looking at the substrate binding mode of the enzyme, they were able to identify potentially important secondary sphere residue that would then allow them to generate a mutant that can actually perform non-selective halogenation and hydroxylation. So I think that was the most advanced work that they've done up to date in terms of converting halogenase to hydroxylase. Moving on to another family of dioxygenase. This time we'll take a look at the Rieske dioxygenase. And this is characterized by the presence of a Rieske center here that contains 2 iron, 2 sulfur motif. And then there's a conserved aspartic group here that bridges the 2 histidines. And this is thought to be important in assisting the electron transfer between the Rieske center and the non-heme center. And the non-heme center is where the catalysis would happen. The Rieske dioxygenase is first identified in the degradation of aromatic compounds by Pseudomonas, and eventually this was found to be a 3-component system, naphthalene and toluene dioxygenase, which essentially perform the hydroxylation of the aromatic ring to give the following product. And in terms of application of the toluene dioxygenase in synthesis, Thomas Hadlicki actually have done a lot of work in using this cis diol product in synthesis. So you can refer to some of his work if you're interested in some synthetic applications. And I've mentioned that this superfamily contains 3 components. So, and this consists of a flavin-dependent reductase, a ferredoxin, and then a terminal oxygenase. And all these 3 components would participate in essentially an electron relay. And this starts by reduction of the reductase domain by consumption of a molecule of NADH or NADPH. This would then turn the reductase domain to the reduced form. And then the reduced form of the reductase would then reduce the ferredoxin, which in turn reduce the oxygenase, which then becomes the active species in your hydroxylation reaction. The mechanism for the aromatic dihydroxylation has not been elucidated in full details, but I think there are some evidence that leads to the following postulated mechanism. So the catalytic cycle would again start from the following iron-free species, similar to what you've seen in the P450 and the alpha-KGs. Similar to the other enzymes, the oxygen molecule will only bind to the iron center only after substrate is bound and iron is reduced. So here, after substrate reduction, there will be a reduction of the iron center, and then proton transfer, and then activation of dioxygen. And this is proposed to lead to the formation of the following bidentate hydroperoxide species. And this actually has been observed by X-ray crystallography. And from here, I think two pathways can be conceived. The first one, which is drawn here, would happen through a coupled O-O bond cleavage and substrate oxidation to generate the following iron oxo species. This would then go on to undergo a radical recombination to give the diol product. And then an alternative mechanism would happen through decomposition of this hydroperoxide species to give the following iron 5 oxo compound. This would then undergo a radical reaction with your substrate that would eventually become your diol species. This would, the diol product would then get released from the active site through protonation and electron delivery to the iron center, and this would regenerate the ground state for your catalysis. I should also mention that this Rieske oxygenases can be highly challenging to study because first they contain multi-component. In general, multi-component proteins are not as easy to study as self-sufficient enzymes. And also the iron-sulfur cluster tend to be oxygen sensitive, and I think this holds true for any iron-sulfur cluster that you would find in nature. And I think the other challenge is that the Rieske oxygenases don't have any distinct chromophore. That makes it hard for spectroscopic studies. Okay, so I've shown reaction of Rieske dioxygenases in hydroxylation of aromatics. And in the next slide, I'll show some other types of reaction that can be performed by other members of the Rieske dioxygenases. On the left, we see aromatic nitration in the form of converting an amine group to a nitro group by an enzyme called PRND. This paper in 2005 showed that PRND can actually accept several other substrates, and this has allowed the authors to study the mechanism of the reactions, and they were able to observe the presence of various intermediates in the reaction, such as the following hydroxylamine and then the nitroso species that would then go on to become the nitrated product. So, in a sense, you can see that the oxygen atom transfer happens sequentially. Then I'll quickly draw your attention to a rather interesting reaction that's catalyzed by several different members of the Rieske dioxygenases. This can be found in the biosynthesis of the prodigiosins. This molecule might have been covered by failed in vitro cycles, but I'm not sure. But in any case, the strategy that nature employs in the biosynthesis of these natural products is pretty interesting because nature takes this common precursor that contains a linear chain called unbezel prodigiosin, and through the action of several distinct enzymes, this precursor can be cyclized in selective fashion to generate different types of macrocycles. To date, there have been at least 3 enzymes that have been catalyzed to perform distinct cyclization reaction, namely Rieske, MCPG, and MARG. And the reaction is thought to happen through hydrogen atom abstraction from one of the CH bond within the linear chain, and then this radical species would then undergo a radical recombination with your parole ring to form the microcyclic ring. Okay, I've covered several different classes of oxygenases, so I'll quickly go through radical SAM reactions that utilizes iron-sulfur cluster. So there are two important cofactor components. So you have S-adenosylmethionine and iron-sulfur cluster, and within the enzyme active site, this form the following complex, where the amine group and the carboxylate binds to the iron in this bidentate motif. And you can see from this crystal structure that sulfonium also has some interaction with the iron center. And the general mechanism of the reaction is the following. Essentially, this enzyme family is responsible for radical generation. Starting from this iron-sulfur SAM complex, there will be an electron transfer that reduces the iron center to the following species. This would then undergo a homolytic cleavage to break the following CS bond that would then generate methionine and the following 5-deoxyadenosine radical that can perform several types of reactions. And I thought I'll quickly go through several energetic considerations for radical SAM enzyme reactions. So it's been estimated that the reduction potential of free SAM itself is about minus 1800 millivolts, and the reduction potential of the iron-sulfur cluster is only about minus 500 to minus 600 millivolts. So based on this energetics, if you just calculate the subtract one from the two, you'll find that radical generation by simply coupling reaction between free SAM and iron-sulfur would be unfavorable. So it's been thought that this binding motif can actually lower the activation energy for the homolytic cleavage. And I also want to discuss very quickly the selectivity considerations for the CS bond cleavage. Look at the structure of SAM itself. There are multiple CS bonds that potentially can be broken. And it's been thought that nature is able to break the following CS bonds selectively by orienting the SAM group in such a way such that the following CS bond would have a direct orbital overlap with the iron center. So in the sense, there will be a direct electron transfer to the anti-bonding orbital of the CS bond that would then promote its selective cleavage. Okay, things that radical SAM can do. This is just some examples. One example can be found in the biosynthesis of biotin. There's an enzyme called BioB that's responsible for sulfur insertion to generate the following heterocycle. The mechanism of the reaction is the following. So starting from SAM, you react it with the enzyme, you will generate the deoxyadenosine radical. And this species can then perform hydrogen atom abstraction of the metal group. In the active site, the enzyme itself also has a sacrificial 2-iron to sulfur cluster. And the radical species here can then attack the sulfur atom within this cluster. And then another deoxyadenosine radical generation event happens, and then this would then perform hydrogen atom abstraction from this carbon. And then this would then do another CS bond formation to generate the resulting 5-membered heterocycle, and also lead to the degradation of your iron-sulfur center. The same type of reaction can also be observed in the biosynthesis of lipoic acid, where so this paper here actually has some pretty cool crystallographic evidence that this reaction is happening. So they were able to trap several intermediates within this reaction. And then finally, I thought I would highlight a pretty cool reaction that's found in the tunicamycin biosynthesis that's catalyzed by a radical SAM enzyme called TUNB. So in this case, the 5-deoxyadenosine radical can abstract a CH bond from here, and then the resulting radical can then do a radical decombination with the following enol ether to generate this important intermediate in the digestive of clinicamycin. Okay, I have about 40 minutes left. Good timing. So I thought with the remaining of the time, I'll cover artificial Metalloenzymes. So artificial Metalloenzymes, I guess we can define it as unnatural enzymes that you get if you insert unnatural metal cofactor that's also catalytically competent in a protein scaffold that typically does not have metal cofactor. And there are several strategies for incorporation of unnatural metal cofactors, and this has been illustrated in the schematic here. So for A, you actually would have a covalent bond between your unnatural cofactor with nucleophilic residues with in the scaffold. This can be achieved by reacting with like a reactive cysteine or serine, or even unnatural amino acid like acetophenylalanine. And then you can also anchor your unnatural cofactor by taking advantage of supramolecular anchoring. So this typically exploits certain substrates that have high affinity to certain protein scaffolds, and we'll see that later on to give you better understanding of what this means. You can also anchor unnatural cofactor by just simple additive bonding to histidine residues in the active site. And then finally, you can also do metal substitutions of metal-containing enzymes. And this is typically done with porphyrin-containing enzymes. I've pointed out several reviews that you can consult to if you're interested in more details of some of these strategies as well as artificial Metalloenzymes. And then I thought I would give a quick historical background on how this field comes into being. And the first demonstration of catalysis with artificial Metalloenzymes was performed by Whitesides in 1978, where he showed that if you make the following rhodium complex that's tethered to the molecule biotin, you can actually anchor this complex to avidin, and you can use the resulting complex to perform catalytic hydrogenation of the following substrate. Now the EE that they get is just moderate, and the turnover number is not great, but this is initial proof of concept that really triggers the development of the whole field. And also I thought I would give a quick primer on biotin and avidin or streptophytin. So biotin itself, structure is given here, and avidin is actually a protein that is found within the avian species, I think. It was found to bind biotin with very high affinity, and later on it was observed that there are also proteins within the prokaryotes that have the same type of properties. In this case, it's streptophytin, which is capable also of binding biotin with a KD of approximately 10 to the minus 14 molar. So it's a very high affinity for biotin. So essentially once you have the two together, your biotin would be bound very tightly to the protein itself. Both avidin and streptophytin are tetrameric protein, as shown in the schematic here, as well as this crystal structure. And I think one of the cool things about this complex is that the resulting chain that extends from your biotin actually would extend out to the heterodimeric interface, and this is actually a nice pocket where catalysis can happen, and that's how people are able to take advantage of this property for artificial Metalloenzyme. So after that proof of concept by Whitesides, the field actually lies dormant for a while until the late 90s, where there was the following paper that revisited the work by employing a different type of ligand to the rhodium center. And the authors found that, again, you can get moderate EE, but the turnover number is not any improved. And in the meantime, Rieske also decided to explore different types of artificial Metalloenzymes for asymmetric hydrogenation. And in this case, he decided to try using a protein called papain, which has a reactive cysteine in the active site. And the cysteine can then do a nucleophilic addition to the following maleimide, which has been tethered to the following rhodium complex. But they found that, as quoted here, the EE values for the reaction is not really good, and they never really proceed any further from here. And so I guess this field was kind of stuck for a while until Tom Ward came along and then did a rather systematic studies on the system. And I think the biggest, the key development that he found was in swapping out apodin for streptophytin, and this was found to really help in getting much better conversion and in selectivity. And I guess throughout this period, the tools of molecular biology was also developing really rapidly, so it's a lot easier for different labs to generate, to do site-directed mutagenesis, to generate different variants of protein of interest, and I think that this really helps in this systematic study. So in this case, they generated a library of streptophytin variants by performing mutagenesis at residue S112. And at the same time, they also synthesized these different types of phosphine ligands that tethered to biotin, and then essentially they performed a combinatorial library screening. And from the screening, they were able to identify several systems that can actually give you more than 90% EE in the reaction for either R or S enantiomer, depending on what you want, and also very high conversion to the product, which I guess this would be about 100 turnover. And once they identify this working system, then they realized that now you can simply swap out the metal complexes to other ones if you want to perform different types of catalysis. And they have been able to show several pretty nice studies for different types of reaction. So on the top here, I've shown an example that they've done in doing Suzuki cross-coupling by using artificial Metalloenzyme that contains the following palladium complex. And they found that they can get about 90% EE with 50 turnover to the following product if they employ the following system that has two mutations within the streptophytin. They also generate a homology model here that helps explaining how the reaction would happen. And they've also done some work in the area of CH activation. This was actually done in collaboration with the Rovis group at CSU at that time. So in this case, essentially, Tom Rovis uses Tom Ward's streptophytin platform to develop asymmetric variant of a reaction that he's been investigating for a while in the following, I guess, what would you call this reaction in?
What would I call it?
Yeah. Sure, okay. CS alkylation. And then I think more recently they were able to adapt this platform for ring closing metathesis. And I think the one breakthrough that they were able to achieve here is that they were able to also couple it with in vivo directed evolution to improve the properties of the enzyme. And some of the key discoveries that they performed in this paper is that they fuse this streptophytin mutant with a protein called ONC8, and this would allow the resulting complex to be directed towards the periplasm of E. coli. And it's known that the periplasm typically has much lower concentration of glutathione, and glutathione can be detrimental for your metal species because of the free thiols. And by having this system, they were able to perform the reactions in living E. coli cells. And if they couple it with the following reaction system that generates a fluorescent product, they can quickly assay the reaction by the amount of fluorescent that they're generating in the reaction. And that way they can quickly identify mutants with improved property. And I've shown here the overall workflow that they perform. So they would generate a mutant library of their streptophyton mutant. They will express everything in 96 well plate. And once they get it to the working plate, they would spin down the cells, and then they would suspend the pellet in the cofactor buffer. And at this point, they also introduced the biotin ruthenium complex and incubate for 30 minutes. So this would allow your biotin complex to be bound to the streptophyton mutant, and then spin down the system once again and discard the supernatant to get rid of any excess biotin ruthenium. And then finally, they resuspend everything in reaction buffer and add their substrate, and then they can assay for their metathesis by looking for improved fluorescence. Now, I should mention that Tom Ward is not the only person that has been highly instrumental in the field of Metalloenzyme. The Lewis lab, now in Indiana, has also been a very active player in the field, and they chose a different system for their construction of their artificial Metalloenzyme. In this case, they chose the pro-oligopeptidase protein scaffold for their artificial Metalloenzyme constructions, and there are several reasons as to why they picked this protein scaffold. So this protein family is cylindrical in shape and also has a large internal cavity that will be suitable for cofactor anchoring. And here they decided to anchor their artificial cofactor in a slightly different fashion. Here they will utilize a unnatural acidophenylalanine in the cavity of the protein, and then they would perform a straight-assisted click chemistry with the following alkyne to generate the complex that they desire. In order to do so, of course, they have to introduce the acidophenylalanine motif, and this is done by the Imber codon suppression technique that's developed by Pitschultz. Okay, so by incorporating all of this technology, they were able to generate this type of artificial Metalloenzyme. In terms of the types of reactions that they can do, this is a rhodium complex, so unsurprisingly, you can do several different types of carbon transfer chemistry. You can, of course, do cyclopropionation, and they've shown that they can get pretty good EE for cyclopropionation of a range of styrenes. And in this case, so the R group here is actually arrow group, so it's a push-pull donor receptor carbene. You can also do CH insertion, and the yield and EE is not optimal, but I think it's a good starting point and good proof of concept. And they've also shown that they can perform directed evolution with this system so that they can quickly generate different types of variants to look for improved properties. And this is a schematic that illustrates how they do it. So they perform error-prone PCR to generate random variants of the specific domain of the proto-oligopeptidase. They would then ligate this part back to the plasmid to generate different construct that would allow expression for different mutants of their proto-oligopeptidase scaffold. They would then introduce that to E. coli host that also contains the necessary construct for introduction of acidophenylalanine. Grow everything in 96 well plate, and then I should also mention that the proto-oligopeptidase that they employ also comes from a thermophile. So the proteins that they get actually is a thermostable protein, so it can withstand lots of heat, and they're able to take advantage of this property in the step where they would heat the system so that they would essentially destroy any other proteins that has been expressed by E. coli. So at the end of the step, ideally you only get your polyoligopeptidase as the active proteins. Okay, so once you do this part, you then transfer the lysate into a microtiter plate and then introduce your cofactor. Now here they introduced a saccharose peat that contains a terminal azide group that would then pluck out any unreacted strain alkyne to minimize any background reaction. And then finally they centrifuge and filter, and the system would be ready for reaction. And then you simply introduce your substrates. And then once the reaction is done, assay everything with HPLC and look for mutants with improved reactivity. Okay, moving on to different types of scaffold. Rufus have had a lot of success in using a different protein scaffold for artificial metal enzyme. In this case, his group employed a protein called LMRR, which stands for Organometallics multi-drug resistant regulator. This is a homodimeric protein, as shown by this cartoon here, that has a large hydrophobic core in the center. And they were able to take advantage of this hydrophobic core for as essentially active site for the artificial metal enzyme. And the first generation of their platform is the following. They use A-burst suppression methodology to introduce amino acid that contain this bipyridine side chain. So this would then bind copper salt. And they showed that they can use this platform to do a asymmetric 1,4 addition to this of indole to this molecule with good conversion and decent EE. And then in their second generation, they decided to do away with the unnatural amino acids and decided to take advantage of the fact that the hydrophobic cavity actually contains two tryptophan residues. And then the thought here is that they would introduce the following phenanthroline ligand that would then have a pi-pi stacking with the two tryptophan residues and have it bound in the so-called active site. And using the system without having to introduce any unnatural amino acids, they can perform similar types of reaction with comparable conversion and EE. I should also highlight several other types of anchoring strategies that people have employed, even though with less success. So here, you can take advantage of the following phosphate to do selective covalent bond formation with a serine residue in protein called glutinase, which is, I think, a serine hydrolase to generate the following complex that you can use for asymmetric hydrogenation. And people have also explored the use of chymotrypsin as scaffold for artificial metal enzyme. In this case, an alpha-chloroketone molecule that's tethered to the following Krebs beta catalyst can react selectively with the histidine in the active site of chymotrypsin. Okay, we've talked about covalent bond formation. I thought I would also highlight a metal substitution strategy that was recently explored a lot by the Hartwig group. So in this case, they decided to investigate the use of myoglobin in carbon transfer reactions. So myoglobin is a heme-containing protein that has, so it has the same protoporphyrin 9 cofactor with iron center, but instead of cysteine like the B450s, myoglobin has actual histidine ligation. So the idea here is that they want to swap out the iron porphyrin to other metal containing porphyrins. And in order to do so, they generate the following workflow. So they would express the myoglobin protein under low iron expression condition. You can do this by simply removing iron from the media component in your growth conditions. So at the end of the growth cycle, essentially you'll get a myoglobin that's not loaded with any heme cofactor at all. And once you purify your apoprotein, so apo means protein that does not contain any cofactor. Once you purify the protein, you can then incubate that with any metalloporphyrin of your choice to generate different types of metal substituted myoglobin. And they then decide to explore all these constructs for the reaction. And they found that if you put iridium porphyrin with the metal actual ligand, this actually performs the best in the following carbon insertion CH insertion reaction. They can get pretty good turnover. Well, there's no EE here, but they can get pretty good turnover. And prior to this work, I think people have not shown that you can do CH insertion with any types of metalloproteins. So once they identify this platform, they then decide to perform the genesis to show that you can quickly alter the EE profile of the scaffold to get any of the enantiomer that they want. So for example, if you go up here, you get one enantiomer through several mutations, and then if you do another set of mutations, you can generate the other enantiomer of the following product. They also showed that you can do other types of carbon transfer reactions, including cyclopropionation of olefin that contains alkyl chain. And prior to this work, cyclopropionation with Metalloenzymes was limited to just styrene type of substrate. So this was a big improvement at that time. And after they disclosed this work, later on they were able to improve the reactivity by using a different scaffold. In this case, they use a protein called CYP119. This is a P450 from a thermophilic organism. So naturally it has a cysteine ligation, but they were able to swap out the cysteine to alanine to free up that coordination space so that they can introduce the iridium metal porphyrin 9. And they show that now with the system you can do intermolecular CH insertion to get decent yield and EE for this product in particular. Okay, so those are artificial metal enzymes, but I guess one question that we might ask is whether this metal substitution is actually necessary to get the reactions that you want. So I thought with the remaining of the time, I'll highlight some of the work that has been done in the Arnold lab that was actually able to do the same type of transformations without having to introduce unnatural cofactors in the reaction, especially for carbene and nitrogen transfer. So you might recall the traditional P450 mechanism, and you might realize that you can easily adapt that to a carbene transfer reaction. So you can have either a diazo compound or a azide. You can decompose those substrates to generate either a carbene or nitrene that can then be a reactive species for many different types of carbene and nitrene transfer reaction. I should also mention that there have been precedents from organometallic literature that this reaction can be done. In 1995, Catalytic, when he was at UT Austin, showed that you can use the following iron porphyrin to do cyclopropionation of styrene with really high turnover. And Ron Breslow in 1985 actually showed that if you use the following iminoiodinane substrate with a rabbit liver P450, you can get about three turnovers for the desired CS amination product. And this formed the basis for this work on artificial carbene and nitrogen transfer. So the first proof of concept was done in 2013 in the Arnold lab, where they showed that by screening different types of P450 variants, they were able to identify one particular variant that they called P450BM3cis P438S that can get the following cyclopropane product with very high cis-trans ratio and high EE. I think this cis-trans ratio is particularly remarkable because usually when you do normal cycle carbonation, the trans product would predominate. And this shows that the advantage of having that protein scaffold for the carbon transfer reaction. And later on, they improved the system by mutating that actual cysteine ligand to serine. This actually was shown to improve the reactivity for carbon transfer while maintaining the cis trans and ED profile of the reaction. And I think the later work in the Arnold lab, when they used P450 variants for carbon nitrous transfer, typically would have this P411 enzyme that has that cysteine to serine mutation. They showed that you can also do NH insertion, unsurprisingly, and a range of anilines with several different types of diazo compounds. And in terms of nitrogen transfer, in 2013 they showed that you can do an enantioselective intramolecular CS amination using the same P411 EM3 cis. This proceeds in about 400 turnover number. And again, with the same system, you can do sulfamidation enantioselectively to get the following type of product with pretty good turnovers. And I should also mention that the Fassan group at Rochester has also developed very nice complementary methods to do the same type of reactions by using myoglobin scaffold. Okay, moving on to probably more challenging reactions. In this case, we'll take a look at several examples on CS functionalization. This year, the Arnold group showed that you can actually do enantioselective CS insertion. This is done by starting from a P411 variant, and initially they found that the initial scaffold that they investigate would get only about single-digit turnover number for the desired CS insertion product. And through, I think, 15 rounds of directed evolution, they were able to identify an enzyme variant that can do the desired intermolecular CS insertion with very high turnover number, about 2,000, with very good ED in the, I guess, high 90s, which is, I think, pretty good. And then they also showed that this transformation actually has a rather broad substrate scope, ranging from insertion to benzylic CH bond, to allylic CH bond, as well as alpha or CH bond that's alpha to nitrogens. And then in terms of nitrogen transfer, you can also do intermolecular CH amination. This was done in 2017 in this Nature Chem paper. This showed that you can do benzylic CH amination if you use tosyl acid as your nitrogen source and P411 variant that they call P411 CHA. They show that this reaction can have pretty good substrate scope with very high EEs. And I think more recently, I think this is a pretty remarkable demonstration of the power of Metalloenzyme for reactions that are hard to do using traditional small molecule catalysts. So they found that they can do reduce-selective intramolecular CH amination by using the following nitrogen precursor. They found, I think, 3 different P411 variants that can do selective formation of different ring sizes of the following amide, ranging from beta-lactam to a 5' ring to a 6' ring. And I think this type of selectivity would be really hard to achieve using your traditional small molecule catalyst. So I think that's the end of the lecture. So I hope that I've given good introduction and overview of the types of reactions that you can do with natural and artificial metal enzymes, and I'll be happy to answer any questions.